What if the key to understanding Alzheimer's isn't just about what's being lost, but about a process that's actively going wrong inside the brain? Today, we're diving into a really groundbreaking discovery that shifts our focus from the plaques and tangles we always hear about to the very electrical signals that build our memories in the first place. Okay, so first, let's just appreciate the scale of this challenge. Alzheimer's isn't just about being forgetful. It's a deep, fundamental breakdown of the brain's most complex circuits, and it affects everything from the tiniest cell all the way up to how we navigate our own world. And right at the center of these early memory problems is this one tiny but incredibly powerful part of the brain, the hippocampus. Figuring out exactly what's happening in there, well, that's absolutely crucial to solving this puzzle. All right, so here's our game plan. First, we're going to look at how a healthy brain builds its own internal map of the world. Then we'll get into the main discovery, how that whole process gets corrupted by Alzheimer's. And finally, we'll talk about what this all means for the future. So let's start with this amazing navigation system that we all have built right into our brains. I mean, think about how you can get up in the middle of the night and walk to the kitchen in the dark. That right there, that's your internal GPS at work. So, inside your hippocampus, you've got these incredible neurons called place cells. You can think of them like little pins on a Google map in your head. As you move around a room, different cells fire to mark different spots, creating this living, breathing map. But just making the map, that's only step one. The really cool part is what the brain does next to make sure that map sticks around. I mean, a map's only useful if you can actually save it and look at it later, right? And that brings us to this critical process for turning a quick experience into a memory that lasts. It's like hitting the brain's save button. So when you're resting, or even sleeping, your hippocampus doesn't just switch off. Oh no, it gets busy. It starts rapidly replaying the exact same sequence of place cells that just fired when you were exploring. It's like this super fast mental rehearsal, the brain's way of practicing what it just learned so it can lock that memory in. And when I say fast, I mean unbelievably fast. This replay can happen up to 20 times faster than the original experience itself. And this just shows you that time compression in action. A 5-minute walk through a new building could be neurally replayed and saved in just a matter of seconds. It is a crazy efficient system. So there it is, the healthy, elegant process. You experience something, you rest for a bit, your brain replays the pattern at super speed, and boom, the memory gets locked in. It's precise, it's structured, and it is absolutely vital. Okay, so if that's how it's supposed to work, what exactly goes haywire in a brain with Alzheimer's? This is where the new science gets fascinating, because the answer was not at all what anyone expected. Now get this. When researchers looked at mouse models of Alzheimer's, they found something stunning. The number of times the brain tried to hit save, the number of replay events, was exactly the same as in healthy brains. This tells us something huge. The brain is still trying. The will to save the memory is still there. And this is the light bulb moment. The problem isn't the number of replays, it's the quality of them. The replay signal itself is all scrambled. Instead of that clean, orderly sequence, A, then B, then C, it's a jumbled, chaotic mess. It's like trying to read a sentence where all the words are on the page, but they're in a completely random order. The information is technically there, but the structure that gives it meaning is just gone. And this quote from the researchers at UCL really just hits the nail on the head. It's a total paradigm shift. We're not looking at a system that just stopped working, we're looking at one that is actively messing things up. So what happens when that crucial save signal is scrambled? What are the actual real-world consequences of all this neural chaos? Well, it's a direct connection. If the replay is a jumbled mess, the brain's map can't stabilize. The memory just fails to get saved properly. So in the study, this meant the mice would forget which parts of a maze they'd already been to, just going back over and over again. And that behavior, it's a striking parallel to the disorientation a person might feel when they get lost in their own familiar neighborhood. But here's the really important part. When you can pinpoint a problem this specifically, it does more than just explain a symptom. It shines a huge spotlight on a whole new path forward. And this is where it all becomes really hopeful. This discovery just blows the doors open for new ideas. I mean, imagine being able to detect this signal scrambling long before any major damage shows up on a brain scan. And for treatments, the target completely shifts. It's less about just clearing out junk and more about developing therapies that could actually help restore the proper timing and structure of these memory signals. So, the big takeaway here is a fundamental shift in perspective. The ultimate goal may not be just to stop memories from fading away, but to actually repair the biological machine that builds them in the first place. This discovery gives scientists a real functional target to aim for, offering a new and very tangible reason for informed hope in the fight against Alzheimer's.